Economic importance. Penyakit merah, which has been known in Malaysia since 1938, was identified as Tungro in 1965. The mentic disease of Indonesia is also identified as Tungro. Tungro is commonly found in Bangladesh and India. In India, it is seen in states of West Bengal, Kerala and other parts of India. Tungro is one of the most widely distributed and most destructive diseases in tropical Asia. The loss was estimated during 1940 as 30% or 1.4 million hectares annually. In Thailand, a severe epidemic occurred in 1966 affecting more than 3 lakh hectares. An outbreak of Tungro in 1971 affected hundreds and thousands of hectares in Philippines. Symptoms: infection occurs both in the nursery and in the main field. Plants are markedly stunted. Stunting is more severe on susceptible varieties and slight on more resistant varieties. Leaves show yellow to orange discoloration and interveinal chlorosis. Yellow discoloration is commonly seen in Japonica varieties, while Indica varieties show orange discoloration. Yellowing starts from the tip of the leaf and may extend to the lower part of the leaf blade. Young leaves are often mottled with pale green to whitish interveinal stripes, and the old leaves may have rusty streaks of various sizes. The plants may be killed if infected early. Tillering is reduced with poor root system. The infected plants have few spikelets and panicles are small with discolored grains. Tungro infected plants can be chemically identified by Lodin test. 10 cm long leaf tip is cut in the early morning before 6 am and dipped in a solution containing 2 grams iodine and 6 grams potassium iodide in 100 milliliters of water for 30 minutes. Tungro infected leaves show dark blue streaks. Etiology: it is a composite disease caused by 2 morphologically unrelated viruses, rice tungro bacilliform virus, RTBV, and rice tungro spherical virus, RTSV. RTBV has a bacilliform capsid or bullet-shaped particles, 130 by 30 nanometers, made up of a single piece of coat protein of MW36K and a single molecule of circular dsDNA of 8.3 kbp. RTSV has a isometric capsid, 30 nanometers in diameter, comprising 2 to 3 polypeptide pieces and a single piece of polyadenylated ssRNA of about 10 kbp. Solar, yellow or orange. Discoloration. 2 types of virus particles are associated with the disease. Bacilliform particles cause majority of the symptoms of the disease. Spherical particles help in the transmission of bacilliform virus by the green leaf hoppers. If the bacilliform virus particles are alone present in the rice plant, they will not be transmitted by the leafhopper vector. Disease cycle: the virus causes severe damage only in area where the host plants and the insect vector multiply the year round. In the areas where the rice is not grown continuously, collateral hosts, especially wild rice, are probable sources of inoculum. Stubbles of infected plants from the previous season also serve as a source of inoculum. Grassy weeds such as Eleusine indica, Echinocloa colonum, Echinocloa cruzgali may be infected occasionally. The leafhoppers Vis, Nuphatetax virescens, N. nigropictus, N. parvus, N. malayanus and Racelia dorsalis transmit the virus in a non-persistent manner. Management 3/4 summer deep plowing and burning of stubbles. 3/4 destroy weed hosts of the virus and vectors. 3/4 grow disease-tolerant cultivars like MTU 9992, 1002, 1003, 1005, Suraksha, Vikramaya, Bharani, R36, IET 2508, RP 4-14, IET 1444, IR50 and Co 45. 3/4 control the vectors in the nursery by application of carbofurum granules at 170 grams, cent, 10 days after sowing and at 10 kilograms. AC in main filed 3/4 spray monocrotophos at 2.2 milliliter. Or phosphomidin at 1 milliliter. Or ethofenphos at 1.5 milliliters. Or neem oil at 3% in the main field 15 and 30 days after transplanting to control leaf hoppers. 17.